All right, in my talk, I would like to cover the key issue which was leading the research in memory mechanisms for more than 1 century. The issue of how memory is laid down, how it is consolidated, a different term. And some new ideas which appeared at the end of the last century, how it can be also reconsolidated. So what is going on when memory is retrieved? I will focus on the history, importance, the fact that these mechanisms appeared to be universal across animal kingdom, so like mechanisms of biophysical processes in the cell membrane or mechanisms of genetic modifications and gene encoding, the memory and plasticity is very universal. And on some unresolved issues. I will swap between covering the broad review of what is going on or known today generally, and our own research, which we were doing during the last 40 years, and it will guide my talk. And finally, I would like to focus on some unresolved issues in the hope that that might interest you and you might probably try to get involved in some of these new studies in the future. Jim Watson, when he was 60, he had a 60th anniversary. He wrote a kind of a note in the journal Science, where he said that the main unresolved issue which is left in biology is how the brain works. Let's hide the bar. The main unresolved issue in biology is the brain. And then he said there are two most important questions about the brain: consciousness and memory. Number two. But consciousness, he said, is still difficult to study. So we should start and focus on the biology of memory. And it was not only a statement globally, but he restructured the work of his own institute, the famous Cold Spring Harbor laboratory, which was a mecca of molecular biology, and he led this research with many Nobel laureates coming from this research, and switched to refocusing the work of laboratory on neurobiology with a particular focus on memory at the molecular, cellular, and behavioral levels in different species, in Drosophila, in mice. Why it is so important? In textbooks of psychology at the beginning of the 20th century, the chapter on memory usually started with the phrase like that: memory is a property of a soul. That meant that this is the essence of a soul, of what makes human a human being. It is in the center of individual soul. And as a famous American historian of science, Ian Hawking, wrote in the book Rewriting the Soul, what happened at the end of the 19th century, that a new science of memory has been created in order to secularize the soul, to make it accessible to the study. It happened at the border between the end of the 19th century and beginning of the 20th century, and many scientists were extremely interested into making the concept of memory material and biological. One of them was German scientist Richard Semon, who worked in Jena. He was a student of Ernst Haeckel and medical doctor, and he wrote a kind of a manifesto, which was published in 1904, and it was called Die Mnem. The central idea of the monograph was that there is a material trace of memory formed in the nervous system. And the four essential characteristics of this material trace, which he called the engram, that these engrams should produce long-lasting, enduring changes in the brain which result from each experience. Number one, the behavioral manifestation of this trace of memory has to appear through interactions with the retrieval cues which come to call this memory back. And he called this process ekphory. The content of engram should mirror, reflect, represent the events during encoding, and they can be retrieved during the recall ekphory. That makes this engram a working instrument of connecting the past to the present. And finally, which is very important, that these engrams should exist in dormant states. That means silent state, but they're kept in the nervous system between the encoding and retrieval phases. That was a theoretical proposal which inspired many scientists after Semon to look for the traces of memory, to search the engrams. That was the title of one of the famous papers of Carl Leschly, who devoted more than 30 years to the search of these traces of memory. But other pioneers, whom I would like to show now, started doing this not on animals like Leschly did, training rats in the different mazes, but from humans. And that was a real transition from the human memory, human soul, which resulted finally at the end of the 20th century in molecular mechanisms and genes of the genome. So it was a radical reduction, much more radical than a reduction of life to DNA. The science of memory emerged in the third quarter of the 19th century when philosophical speculations about how individual memories are stored were transformed into a program of research, mainly by three directions of research and three pioneers. Hermann Ebbinghaus, who was the first to study normal human memory in psychology by experimental psychological terms. Théodule Ribot, who worked with in Paris with the patients with different neuroses and studied the disturbances of memory with the psychopathology of memory, and from that derived many cues about how memory works. The famous Ribot law, the law of regression of memory during aging. And finally, Sergei Korsakov in Russia, who was extremely interested also in the mechanisms of memory, and being a psychiatrist, he studied different patients who came to his clinic with memory disturbances after various types of conditions, starting from fever and viral infections, going to poisoning, which led to loss of memory, or what he was famous for, chronic alcoholism, drug intake, where the chronic use of alcohol led to degradation of mammillary bodies and severe amnesia, which was called Korsakov syndrome. I will focus now only on one of these programs. The program which led to the idea of memory consolidation, and as I told, this program guided the research in memory over the 20th century. It was started by Hermann Ebbinghaus. Here he is a respected scientist, but when he started, he was slightly over 30 years old. And in Paris, he came across a small book of Wilhelm Wundt, who started experimental psychology, and Wundt wanted to study consciousness. Ebbinghaus read this book, he was excited and thought, why not to try to study experimentally also memory? And he started to work with the memory consolidation using himself as an experimental subject. That was phase 1. And I will switch later on to phase 2. So what Ebbinghaus did, the famous forgetting curve, is that he designed many cards. Each card consisted of a syllable of 3 letters, and he presented them in random order to himself in order to remember, and then tested himself how much he needs to effort to recall these cards or to remember them once more at different times when he repeatedly showed them to himself. And what he found was very striking, that initially, immediately after presentation, he was able to recollect 100% of the cards. And then there was a dramatic drop of the recollection, going to about 30% within 1 hour or several hours. But the rest, this level of the recollecting these cards stayed for a long time. So it became apparent that there are 2 phases of memory. One is going within the first hour after acquiring information, and something important is going on there. Part of the information is lost, but the other part is retained. So there are these 2 phases, the early phase of remembering a lot, and the late phase stable for a very long time. 2 German scientists, psychologists, Elias Müller and Alphonse Pilzecker, working in Göttingen, were very excited with this work of Ebbinghaus and decided to do the same type of experiments on volunteers. So that was a proper study with many subjects. But they not only repeated the experiments of Ebbinghaus, but introduced one very important experimental moment. They presented the data and they used either pictures or syllables like Ebbinghaus. But after presenting these cards or pictures, they made a presentation of a novel stimulus, like, for example, here, some new picture. And they showed in their experiments that when this stimulus is presented immediately or within the first minutes after trying to remember some information, this information is disrupted, it is lost, it is no more remembered. If to delay the presentation of this interfering signal stimulus for a later period, after 15 minutes or so, the information is remembered. So they made a fundamental conclusion. They called this a retroactive interference, so interference back to the initial information. And they came with a conclusion that after something is remembered, we get the new information. It is stored in the brain in the labile form, and it can be easily disrupted by some interfering conditions. But when the time passes and the memory goes to the second phase, it is stable and it is encoded as they believed in the anatomy of the brain and connections. So remember, Simon, the engram is laid down and kept dormant until it is retrieved. They suggested that this process of transition from labile form to stable form should be called consolidation of memory. And since that, we work with this concept, how our memory are stored and remembered, they are consolidated. Immediately, different scientists in different countries got this idea and confirmed it by other evidence, psychological evidence or clinical evidence. For example, McDougall, neurologist in UK, pointed that this is very consistent with what happens with the human amnesia in the conditions of epileptic seizures, large epileptic seizures, when the person gets into the seizure and then recovers from seizures. The person does not remember few preceding minutes before the seizure started. So it is a retroactive interference of the seizure activity in the brain with the memory engram. And very similar things he observed also in professional boxers when they lost consciousness during knockdowns. They do not remember few preceding minutes before that has happened. However, for a long time after this has been confirmed and the psychologists started to believe that memory is consolidated through these two phases, there was not much progress. And the reason was that it was not possible at that time to do any imaging, recording, or invasive studies in humans to experiment with the neural mechanisms of this process, and nothing was known about animals until at the end of 40s, two American psychologists, one of them is Duncan, showed independently that if to transfer what we just described in us, in humans, to rats, and to reproduce this in a slightly modified protocol, for example, not like seizures in humans, but to produce electroconvulsive shock, just putting the electrodes on the head of the rat and giving the shock which produces the seizure-like activity in the nervous system. And if the rat is learning something before that, it is trained, and then to apply this electroconvulsive shock seizures, the rat is not able to remember what it has learned later on, but if to delay the presentation of these seizures like in human conditions for 15 minutes, the memory is retained, so it is consolidated. And it became clear that fundamentally the same process of memory consolidation is going in animals, which opened a venue for different type of speculations of what can be studied in animals. And since that was the beginning of the revolution of DNA and genes and encoding of information in macromolecules, several people started to speculate that the only complex information bearing molecules which are retained in the nervous system for a long time is DNA. So memory, if it is stored for the lifelong events, should be somehow encoded in DNA. So there should be changes in the genome, and expression of memory should involve modification of gene expression. There were not many instruments at that time, but still famous Swedish neurobiologist Holger Hedén started to isolate RNA from the animals which were trained under different conditions, from rats, for example, which were trained to climb with a small narrow path to obtain food and to make a motor coordination. He isolated RNA from the motor nuclei of these rats and showed that in the course of learning, there is an increase in the incorporation of radioactive precursor in RNAs, which means that there is a burst of synthesis of RNA during acquisition of new memory, which tells that it is a activation of gene expression. Similar data were obtained by other groups using labeled leucine and measuring the protein synthesis in the brain. It appeared that whenever the new experience is acquired, within the first hour, this first hour of Ebbinghaus, there is a burst of increase in RNA and protein synthesis, which means that some genes are activated in the nervous system. And absolutely reasonable test to see whether it is just a correlate or it is necessary for the memory to be stored was to apply the inhibitors of RNA synthesis or protein synthesis, which again a number of groups did at the beginning of 60s. And they showed that either actinomycin D, which is the blocker of RNA synthesis, or cycloheximide or anisomycin, which are inhibitors of protein synthesis, when they are injected, and this has been done in many different species, rats, mice, cats, rabbits, etc. When they are injected prior to training, number one, acquisition of memory is not impaired. Animal learns and acquire new information and is able to reproduce it. Second, for a short period of time, several hours, the animal remembers what it has acquired normally. So you can test it and the animal will show that it recollects the situation in which it was before. But after that, after several hours, the memory is lost. it is not possible anymore for the animal to remember what is going on. So the idea was confirmed that when the new memory is acquired, there is increase in the RNA and protein synthesis, which means gene expression. And these proteins are finally necessary to make the memory stable and last it longer than few hours. That was the situation in the middle of 70s, and it was quite clear, but the problem was that biochemistry of that period was not sensitive enough to isolate what are the proteins and what are the genes which are involved in this process. So the research in this area stuck for about 10 years, though it was clear that something is going on, but it was necessary for the new methods of molecular biology, gene cloning, and sensitivity to detect gene expression to appear in the middle of 80s to get back to these fundamental questions, which are here listed in one of the reviews in 80s. So there is a stimulus which produces the release and action of modulatory transmitter. Then the signal through cyclic AMP in this case and protein kinase goes to nucleus, but it is not known what is going there. And it is also not known what are the genes which are expressed and what do they do to modify the synaptic properties in neurons. So that were three questions. What genes are induced during memory consolidation? By what mechanisms are they induced? And what are their functions in neurons? At that period, we have started looking for these genes. There were two groups which initiated this work. One was in Soviet Union, and we worked with the Institute of Normal Physiology, Institute of Molecular Biology, and Institute of Molecular Genetics. And independently, there were two groups working in Eastern Europe, in Poland, in Nensky Institute, Leszek Kaczmarek, and in Germany, in Magdeburg, the group of Hans-Jürgen Matthias. And both groups came through different routes to identify the same genes which were shown to be involved in memory consolidation. We initially found these genes through whole body in situ hybridization by looking into the development programs of proto-oncogenes. And these genes were known initially as proto-oncogenes, the genes which are counterparts of viral genes but perform some important functions like cellular differentiation and regulation of cell cycle during normal development. And it appeared to us that these genes are expressed in the proliferating cells, but also expressed in the developing nervous system, which reminded the old idea of Ramon and Cajal that probably the cellular mechanisms which lie behind neural plasticity and memory is reactivation of old mechanisms which are involved in growth of the nervous system. So we decided to test how these genes behave in the adult brain during acquisition of memory, and it appeared that these genes, there were several of them. The most striking was gene C-force. They are silent. They are not expressed in the brain of a normal animal, which is not involved in any new experience, performing just regular normal behavior. But whenever animal is placed in the conditions of novelty and acquisition of new knowledge, new experience, there is a burst of expression of these genes in the nervous system. Going within, RNA is induced within 15 minutes, and protein starts to accumulate within the first half an hour, hour. That means with this first critical period, which was shown by Ebbinghaus to lie in between the labile memory and consolidated memory. We also showed that it's not only that these genes are expressed, and we tried this on different animal models and in different types of learning, classical conditioning, instrumental conditioning, episodic-like memory, etc. But also, if to block the expression of these genes using, in this case, antisense oligonucleotides, the small oligonucleotides which prevent specifically expression of the gene of interest when they're injected. So when they're injected in the brain of animals that learn, and this is the acquisition curve which shows the amount of mistakes which animal is making during the session of training, you can see that exactly like with the protein synthesis inhibitors, blocking just one of the genes, C4s, in the brain during learning has no effect on acquisition of memory. They learn normally. I do not show here, but if to test them 1 hour later, they will show that they remember this. They make no mistakes when they are placed again to be tested in this task. But if to test them 24 hours later in the first 20 trials, which is a reminder of what was before. So they do not learn at this time, but they recall what was known. They are severely amnestic. They don't remember what they learned on the previous day. So not only expression accompanies learning and memory, but it is critical for memory consolidation. It immediately also occurred to us that these properties of immediate early gene expression are very suitable for mapping the memories in the brain, because besides the electrical activity of large amount of circuits, which are denoted here by yellow impulses traveling in the brain, there are some of the ensembles of cells which induced expression of C4s during acquisition, and looking at what are these cells, where they are laid down, how it is done in the whole brain. We can trace the engram, the Semon elusive engram, material trace of memory, which is laid down. This line of research also initiated the answer to the second question, because once you know that the genes which are induced during acquisition of new memory and its consolidation are these genes which became to be known not as proton genes, which was just coincident name initially, but immediate early genes, because they are turned, these are the most rapid response genes known in biology. Within about 250 milliseconds after stimulus arrives to the cell, the transcription starts in the nucleus to synthesize RNA for this gene. And within 3 minutes, the protein first appears in the cytoplasm. Very rapidly inducible genes. So they were called immediate early genes in the analogy to viral genes, which are initiated in the first round of expression of viral infection. So once you know that you should look for expression of immediate early genes after stimulus, which leads to plasticity, you can trace it back. What are the factors which are binding the promoter of immediate early genes? And what happens with these factors? Why they start to induce the transcription? What signals are initiating these factors to become active? What are the cellular signaling cascades which lead from the membrane, from the synaptic signaling to the nucleus to initiate this? And it appeared that one of the important factors was constitutive transcription factor, which was called CREP, and it is present in the cell, but it is unphosphorylated. When it is not phosphorylated, it is not active, and it is not binding the CRE elements, the promoter elements of immediate early genes. In order for it to become active, calcium and other type of signals, as I will show in the next slide, has to enter and converge on different kinases, which are able to phosphorylate CREP and to initiate induction of immediate early genes. Another thing which we found was clear that immediate early genes like C4s are transcription factors. That means that they do not do anything physical in the synapses. When the proteins are synthesized, they are transported back to nucleus to regulate the activity of many genes which are specific to this particular neuron. So if the mechanisms of immediate early gene is a critical bottleneck for memory consolidation, we should observe later on the second phase of memory consolidation, which nobody looked for because it is quite late at several hours after acquisition. Ebbinghaus didn't look, psychologists didn't look. But now knowing that there are two phases, we started looking at whether there is another wave of gene expression in the brain after memory acquisition, and whether it is possible also to block memory consolidation by selectively inhibiting this second wave of gene expression. And indeed, we found that from 5 to 8 hours after we learn something, animal learns something, there is a second large wave of gene expression. And if to inject protein synthesis inhibitors at this stage, you can again erase memory, prevent it from being consolidated. So we came to the two-phase model because of these immediate early genes of memory consolidation in the cell, which gradually became more and more complicated. And I'm showing just one of the diagrams demonstrating how many signaling cascades are involved in regulation of this process in the cell. It is important to know that they are complementary. That means that increasing cyclic AMP level to a certain level in the cell will not trigger immediate early gene expression. Increasing calcium levels will not also produce it to a certain level. But if you combine these two small stimuli converging on the same neuron, you have a superinduction of this immediate early gene expression. This is because these factors combine their effect on the promoter of this gene, triggering it much stronger than the single factor. It also became evident that this cannot be the only mechanism for memory, long-term memory consolidation, because if you will take the expression of immediate early genes and then late genes, the proteins which are synthesized from this burst of expression during learning, they will be retained in the cell for some hours or even days or probably even a week, but then they will be degraded and the memory will be lost. So besides this trigger, genetic trigger for memory consolidation, there must be something more stable, which will maintain now this cell in this differentiated state, which was induced by learning and triggered by transcription factors like C4 and other. And indeed, it appeared through pioneering studies, by the way, initiated in Russia at Moscow State University in 1970s by biochemist ANOKHIN and neurobiologist Tushmalov, that besides induction of gene expression, learning needs to changes in the methylation of DNA and long-term rearrangements in the DNA functions due to acquisition of new experience through different type of enzymes, de novo methyltransferases, and also the chromatin in these cells is changed for a long time through acetylation of histones, proteins in the chromatin, to mimic in a way the process how cells are becoming differentiated again during development. The same story, that something which is going during development and make cells stable to maintain their new phenotype is recruited again during the process of memory consolidation. One of the consequences of this, why it is important, it appeared that a number of diseases which were initially believed to be inherited genetic disorders, which is not able to be cured, like Rubinstein-Taube syndrome, appeared to target the processes of plasticity. So children which are born with these syndromes are not able to learn new things, and it results in mental retardation. And it was believed for a long time that it is inevitable and nothing can be done because it is a genetic disease. In fact, it can be corrected if to improve the particular CREP functioning cascades in the nervous system of these children. All right, with this, I will finish the known part and will go to some new findings which resulted from these studies, and some of them are already established and others are not. By 92, in our lab, we believe that we know more or less what are the molecular mechanisms of regulation of gene expression during memory formation. Of course, we were wrong, because since that, many new things appeared. But we had a feeling that the basic mechanism has been found, and it is necessary to move on and to look into some unknown domains. And at that time, I made a list of 10 strange phenomena which were not explainable by the knowledge of that time. So the facts which were contrary to all our knowledge and expectation. This is the list, and I will go only through a couple of them. We started from the beginning. There were some strange things found in the literature which we started to explore. And here I will introduce one of experimental models which we consistently used for many years. It is very robust model of one trial long-term memory. It is formed in newborn chicks. When the newborn chick is presented with a small bead, it immediately pecks it, expecting that this is a kind of a food. But if to cover this bead with a bitter testing substance like quinine, the chick will shake its head, will vocalize in disgust, and from just one peck, this object will remember this object comparable to all other similar objects of different color, of different shape. It will start to avoid this object comparable to all others. So that means that within a couple of minutes, about 1 minute of training, you can selectively localize the time of memory acquisition, which leads to long-term memory consolidation. And you can go through all the phases and see what happens on biochemically, molecular, neurophysiologically, and neuroanatomically in the brain to consolidate memory. This is presentation, just video. The chicks are presented with the white neutral bead. It is covered with water, so they are not avoiding it. Now we present another bead that was silver, and this is white, and it is covered with a disgusting substance, methylantranilate. Absolutely disgusting, you can see. Now, when you present this aversive bead a day later, you can see that they refuse to pack it and avoid it. If you present the neutral bead, which was not covered with something bitter, you can see they readily pack it. They remember this particular object for a very long time, long-term memory. It is protein synthesis dependent, RNA synthesis dependent. It is known where it goes in the brain of chicks. You can inject the substances exactly into this area, etc., etc. So taking this model, we started to look at a strange thing, which was not a part of the consolidation theory of memory for 100 years. What will happen if already consolidated memory is retrieved and you will try during this retrieval, and the memory was believed to be stably encoded, to interfere with the same type of approaches which you use to interfere with acquisition of memory. So protein synthesis inhibitors, electroconvulsive shock, etc., etc. If it is like PDF file, you retrieve it, watch it, and then close it, nothing happens. But we found in our experiments in force expression something strange. When the animal is trained in a very similar task, one trial also task in mice in fear conditioning, where it is presented conditioned stimulus and then receives a food shock. And then later on is tested by presenting this conditioned stimulus again and remembers that what is happening. During retrieval, there is a very strong expression of C-force in different brain areas. why it is going on if it is already consolidated. What force transcription factor is doing in this episode. To address this, we took the situation first in chicks and then in mice, where we designed the experiments when the animal is presented with the new stimulus for acquisition without any interference. So later on, when we remind showing the same aversive bead, which is, by the way, not covered by beta substance, it is just looking the same. And if to try to inject different substances which are known to prevent consolidation of memory, acquisition of long-term memory, like protein synthesis inhibitors at this time, what will happen? If the memory is stable, nothing should happen. The maximal what should happen is probably if the animal learns a little bit more from the second episode. You will wipe out this surplus, this additional memory. But in fact, it appeared that if we injected cycloheximide protein synthesis inhibitor alone without activating memory, there is no effect on performance. The chick perform at 90% confidence level of retrieval. If to make a reminder and inject saline instead of cycloheximide, so not inhibiting protein synthesis inhibitor, again, chicks, when they are tested later on, show perfect memory. But if to combine these two manipulations, each of them having no effect, blocking protein synthesis inhibitor during retrieval of memory, the old memory, which was already consolidated, can be disrupted. We did this in mice. Later on, group of Karim Nader in Columbia University did this in rats, obtained exactly the same design of experiment. They used anisomycin instead of cycloheximide only, and obtained the same results. And people started to check what is going on here. And it appeared that this works in mice, in chicks, in fish, in crabs, in mollusks, different mollusks, in insects, in honeybees, in C. elegans, in Drosophila, etc. It's again a widespread phenomenon that when memory is retrieved, I will add under conditions of novelty, old consolidated memory becomes labile, and it is possible to disrupt it with many interfering processes. So it has to undergo again the process of laying down back, reconsolidation. I'm showing this just to show this is a this year review of molecular mechanisms of reconsolidation of what kind of animal models. You will not read it here. It is just an overview of how many and what kind of molecular mechanisms are known now for the processes of reconsolidation in different species. The overall conclusion, reconsolidation is very similar in mechanisms, in molecular mechanisms to consolidation, but not identical. A number of things are unique to process of reconsolidation, which are different during the mechanisms of consolidation. But both of them lead to long-term changes. Why it is important? It transforms our traditional understanding of memory. So besides encoding and the process of laying down memory of consolidation, there is a process of making it active, retrieval, and after it is retrieved, it is labelized and has to reconsolidate again and new retrieval. During these periods of retrieval and reconsolidation, memory can be and is updated. So it changes many things, its properties, its substrates, etc., etc. Another important transformation that means that there are two time windows now that memory may be labile and vulnerable during encoding phase and during reactivation. One of the differences you can see here, the process of molecular consolidation during initial memory formation is longer lasting than a process of its laying back reconsolidation. But nevertheless, why again it is important? First, I showed on that table, humans, we undergo the same process. And it was foreseen by Sigmund Freud, who didn't develop it further enough at his time because he didn't have enough experimental material and approaches. But he wrote to his friend Fliess at the end of the 19th century. Как ты знаешь, я работаю над предположением, что содержимое следов памяти подвергается время от времени реорганизации под влиянием свежих обстоятельств, ретранскрипции. He called this retranscription instead of reconsolidation, but it is the same. Принципиально новым в моей теории является тезис, что память закладывается не единожды, а на протяжении ряда эпизодов ее востребования, то есть извлечения. Many times, many traces, multiple traces of the same memory. And here is just a table again from the review of 2025 on many evidences of human reconsolidation in all variable tasks, in episodic type memory, in the picture memory, in the motor memory when humans are learning to do something and you can interfere during retrieval and impair it. So it's again a broad phenomenon. I wrote a letter to the Minister of Science when we discovered this, and they closed our publication for several years, prohibited to publish these results on the quite evident basis, because with these interference, you can erase old memories. And indeed, when we finally published and other groups published, it became very popular. So one of the films you might have seen, where the lover wants to get rid of the memories of his girlfriend and goes to the company which is called Lacuna Incorporation, Memory Management Incorporation, which presents the reminder stimuli, which was part of their common experience under particular treatment of his brain to erase the memories of his girlfriend. But more practical is that what was also evident, and I suggested in this letter to ministry, that it is possible to treat different traumatic memories, like in post-traumatic stress disorders or drug dependencies. We also showed at that years, at the beginning of 90s, that formation of alcohol and morphine dependence goes through the same mechanisms of memory through C4 induction. By showing the episodes which produce this traumatic memory, using the pharmacological drugs, there is a number of them which can be used in humans to prevent reconsolidation. And these attempts are going on since then with the relative success. Not always that works. There are many conditions why the memory can return back, but still. Когда я начал? Это остаток, да? But now I want to go to something new, which we were working in the last several years. Think about reconsolidation in this fashion. Imagine that you are getting some influence interference which makes you difficult to remember new things, consolidate and reconsolidate, like a large dose of alcohol. And after that, you will not remember what has happened at the time when you were drunk. If you remember something at this period, retrieved from memory under these conditions of inability to reconsolidate it, you risk to lose this memory or weaken this memory because it will be not laid down since the mechanisms of consolidation are impaired by alcohol. What it might remind or what it might look like. One of the immediate ideas, this is something which happens during development of Alzheimer's disease. When the mechanisms of laying down new memories, because of many processes, pathological processes in the nervous system are compromised, are impaired. So people are not able effectively and gradually more and more to remember new things, where they left the car, where they left the keys from the flat, what they, whom they met yesterday, etc., names, etc., etc. This is called anterograde amnesia, inability to consolidate new memories. But if you will look through the glass of the reconsolidation theory, you can also add to this that if these memory mechanisms of consolidation are impaired, and at this, on the background of this impairment, the patient will start to reminiscent, recollect some old memories which are retrieved and become labile. They will also have difficulty to lay back. That means that by retrieving old memories, the patient with Alzheimer's disease destroys them because they are not able to get back, to be consolidated, reconsolidated. And that means that it might lead to retrograde amnesia. A second very dramatic symptom of Alzheimer's disease and dementia generally. The people are starting to forget their relatives, their past, who they are. They cannot recognize themselves in the mirror. They lose, like I told, during human memory consolidation, you can interfere with the motor habits, etc. They lose their day-to-day survival habits, how to eat, how to wash their teeth, etc., and etc. So we came with the hypothesis which I called the Sodom hypothesis. Sodom, from the biblical Hebrew, to burn, to ruin, to destroy. Sodom is self-onset disruption of memory. Disruption of memory which is initiated by memory itself, by its activation. If memory is retrieved when its reconsolidation mechanisms in the brain are impaired, it is degraded. Thus, old memory activated in a pathologically altered brain destroys itself, leading to retrograde amnesia. We can compare this to the traditional explanation of why that retrograde amnesia happens, which I'm sure is also true. The traditional explanation, I call it the passive decay of engram. It is because of the development of amyloid plaques, the changes in the disruption of synaptic transmission, and finally loss of many neurons, parts of the engram start to be destroyed. And people are losing the old memories, retrograde amnesia. The features of this process is that it is passive decay of dormant engrams, what Salmon called dormant engrams, which are stored passively, just because the substrate of engram is destroyed. The mode of this amnesia is use independent. It doesn't matter whether you recollect this memory or not, it will be destroyed anyway statistically. The target is passive engram. And the only protection way, if you will look practically, is to protect from neurodegeneration, from the loss of synapses and neurons. There is no other way. According to Sodom hypothesis, there is a second route, which is disruption of active engram when it is retrieved. Comparable to passive route, it is self-destruction of these engrams. It is use dependent. If you not retrieve it, it will be not degraded. The target is not passive trace of memory, but active engram at the time when it is expressed in the activity of neurons which maintain it. And the way to prevent, as I will show you, can be also different. You can interfere with the processes of localization memory during reconsolidation. So we don't have much time left to go briefly through this. We showed that if to inject amyloid in our chick model, beta amyloid, first, number one, it impairs the consolidation of old memory, of new memory. So if you inject it at the time of training, the memory will be at the time of testing destroyed. So what will happen if you will inject it at the time of memory reactivation? We showed that if memory is formed normally and at the time of reactivation, you inject amyloid like protein synthesis inhibitors or NMDA antagonists, etc., it will produce the disruption of normal memory, though injection of beta amyloid alone at this time, or reminder and reactivation with the injection of saline produces no effect, like I showed you before. And this can be shown either at 2 hours test after initial or 24 hours test. At 2 hours, you can see how strong the effect is. Now, what can we do with this? This is the diagram of how reconsolidation works. There is a trace of long-term memory. Then there are conditions which are important, that the memory has to be retrieved under some novel modifications of environment. For example, if I eat for many years with a spoon, and I have this memory, a habit, worked well for millions of times. Injecting the interfering agent at the time when I will use the next time spoon will be not efficient. But if you somehow introduce some novelty in this, tie my hand and make me try to use the spoon in some different way, which was not usual for me before. So these conditions of surprise and novelty will produce first destabilization of old engram, which was used before. And we know that this process is NMDA receptor and calcium signaling dependent. And after the memory is restabilized, it should go to be restabilized. That means reconsolidated. And we know that this depends on the gene expression and epigenetic changes. And as a result of these two processes, the long-term memory under the conditions of normal, when it is retrieved, it is reorganized. What that means for the prevention interference with the reconsolidation. First, you can influence the process of restabilization. And this is what I showed so far before. Memory will be disrupted. You can also do something different. You can prevent novelty conditions for memory retrieval. And in fact, it works in Alzheimer patients. It was found by many clinics, so-called memory clinics, where the patients are treated, that the best way to prevent from the rapid degradation of memory in Alzheimer patients is to keep them under conditions of everything familiar. So to make no novelty. Then the development of retrograde amnesia slows down. And it is obvious from this hypothesis why it is. Because there is no destabilization of memory which is not being able to be restabilized. But the third interesting opportunity is, even with allowing the novelty, to prevent selectively destabilization of memory, since these are specific mechanisms going through specific subtypes of NMDA receptors which are necessary to destabilize memory. You can still retrieve memories under conditions of novelty, but under protection of these drugs, which doesn't make it allow them to become labile. And that means that the deficit here in restabilization will be not evident because they are not labilized, they don't need to be reconsolidated. So to make long story short, we tested this with the inhibitors, antagonists of NMDA, antagonists of NMDA receptors, and showed that when you block the memory at the time of reactivation, if you do this with the injection of antagonist of NMDA receptors, MK801, it can prevent disruption of memory by protein synthesis inhibitors. So it can work as a protector of active memory. And you can do it with the drug which is used to treat Alzheimer's disease, which is a partial NMDA antagonist, memantine. It is one of the 4 drugs which is currently used for treatment of Alzheimer's disease. It produces exactly the same effect, and we believe that through the same mechanism. What are boundary conditions for this hypothesis? I have to stress it. Reconsolidation of memory compared to consolidation occurs not for all forms of memory. And even for those where it happened, it doesn't always happen. It was called boundary conditions. We don't know why some memories are not disrupted at the time of retrieval, but this is the fact, still to be found. Most probably such boundary conditions are applied to memory reconsolidation in Alzheimer's disease. And it is also likely that this process can occur in a restricted window in the development of Alzheimer's disease, when the memory retrieval itself is not compromised too much. So not at the late stages of the Alzheimer's disease, when the patients will have huge difficulties of retrieving the old memories already. I will stop here. Вопросы? Я думаю, I think so. I have another part how it is possible to restore the lost memories, but better to use questions. Можем, наверное, включить свет, да? На английском языке. Да. Да ладно, ладно, молодежь, конечно. Оль.
На английском, ладно. Thank you for your выступление. Talk. I've got a question. It's a bit far from the topic of your talk, but nonetheless, I'm very interested. How are the false memories formed? And what happens on the molecular and cellular levels? Like you remember some event which actually did not happen. Like, how is this formed?
The mechanisms, molecular mechanisms of forming false memories comparable to normal memories. To the best of our knowledge of how memories are formed and consolidated are not different. Fundamentally, if you will look in neurons, in cascades, in genes which are laid down during formation of false memory, they will be the same, just the memory is false. The reason why memory is false is an interesting one. There is one clear story which is evident how false memories are formed. It is due to process of reconsolidation. It has been shown that if people remember some very strong, striking event, which initially in 1960s, 70s, 80s was believed to be stamped in the brain very strongly. It was called the flash bulb memory by two American cognitive psychologists, and they studied these by interviewing people about whether they remembered where they were and what did they do during assassination of President Kennedy. And they did it 10 years after that. And they found that 80% of Americans who were adult at that time were able to point down precisely what happened at that time, where they were, what were the surroundings, from what they heard the news, etc. Until in 1986, a famous American psychologist Ulrich Neisser repeated this type of experiment with his students during the catastrophe of Challenger, where the American cosmonauts were launched. The rocket was launched and it exploded, and they died in front of all the people watching the TV. And it was a great shock, like a Kennedy assassination. And on the next day, he asked his students, the course which he was teaching in psychology, to write down exactly what were the conditions, where they were, and what they were doing when they heard this news. And they put this on a paper. And a year later, he returned back and asked them, the same course of students, to write again what they did. And it appeared that many of them wrote completely different stories, and they were absolutely confident in that was where they were, what they were doing, to the degree that when they were shown their original protocol writings, they were not ready to believe that this is why it happens. I didn't touch many things of memory processing. The strong thing which happens is that when the memories are formed initially, they are then repeated many times, especially if it is important memory, offline. Offline, that means that when the person is not thinking about this, it is going on subconsciously. Or when the person is sleeping during the sleep. And each such cycle of reactivation can lead to enrichment of memory, but it can lead to modification of memory. There is many things to tell here, but briefly, that's it.
Yes. Thank you again, Konstantin Vladimirovich, for the informative lecture. I want to go a little bit in the macro, I think. So to ensure myself, as I understand, the memory is storing inside the neuron, yes? Not in synapse, or this is not very significant in this matter of consolidation of memory?
Both of Pavel Miloslavich and I smiled. The classical hypothesis of memory, which was suggested in neurophysiological terms in 60s by John Eccles and then worked on during LTP studies, that it is a synaptic process. But there are more and more data accumulating that even if you destroy the synapses which were active at the time of laying down the memory, and then allow the neuron to grow a new connection, the memory can be expressed through different synapses, which leads many scientists now, not many, and this type of experiments, Glanzman complained to me that he cannot get funding from NIH for this. But still, there is serious concern that maybe the template for long-term memory is laid down, as you said, inside the cell, and you need synapses to express it in the circuitry of all the brain to get the final outcome. But it can be produced from within. Synapses are finally necessary, but it is not the place where it is stored.
So we can say that every neuron is an n-gram neuron, yes? Because they all, not all of them store.
We cannot say this. Some years ago, we started a systematic project at Kurchatov Institute, which I wanted to perform to answer this question. You can experimentally address it in a simple way. You can systematically test whether all neurons in the brain, or more simplistically, all types of neurons in the brain, different types of pyramidal cells, different types of interneurons of different cellular phenotype, all of them sometime are involved in the process of immediate early gene induction at the time of learning, which means that they were plastic at this time. Or there are some neurons which never do this. We didn't complete it. There is no extensive work that since then was done by anyone. There is another way, by the way, which we didn't do, but it is also possible. We have the double transgenic lines, which allow to accumulate the changes in permanent changes in neurons which are active during particular episode because of the recombination which we introduce in these neurons, and they start to express fluorescent protein which was silent. So if you repeat it many times, you can, during the lifetime of an animal, you can see whether by the time when it is adult and learn many things, all neurons were stayed, or there are some populations which are not. I cannot answer this.
Thank you very much.
Great, thanks. Amazing presentation. I just have one methodological question. I have never worked with CFOS labeling, and in some slides you mentioned this method. And CFOS labeling, as I know, it shows total neuronal activity, but you mentioned learning activity. So how it works, yeah? So if we look in your slides, this neural activity, for example, in cortex, yes, it can demonstrate just reaction of cortex on learning process, but not like exactly learning, yes? So, for example, there are some articles which demonstrate like governing food, for example, food behavior. You can feed rat, yes, and also via CFOS method, you can detect governing energy balance, like food addiction. So methodological question about CFOS labeling, why you like...
It's not only methodological, it can be technical and addressing what you are labeling, but it can be, and it is, a scientific one. Again, I don't know. In 1980s, when we found the expression of CFOS, I had two ideas. First, that CFOS expression is a key factor for memory consolidation. So it is okay signal for the cell to remember something, definitely remembering. Or it is a kind of a competence factor. That means that the cells which are activated within the population, they start to express CFOS as a transcription factor. It puts the cell in some competent state, like in immune cells. But this is not sufficient for memory to be laid down. There is something else which is missing. So not all cells, and we did correlative study with neuronal recording and CFOS expressing, showing that from the neurons from the same areas in rats which learn to acquire new experience, only about 10% of neurons which were expressing C-force at the time of new experience become specialized electrophysiologically by their firing to this new experience. So probably many of the neurons which express C-force are not exactly the memory cells, the engram cells. But the situation is not that simple. First, that idea that C-force is labeling active cells came from the people who didn't know comparable to what we with Kaczmarek and Matis worked in at the beginning, what is going on in the real experimental situations. So it was a kind of a dogma which was not well founded. We know that there are brain areas where there is a strong neural activity. You can record it electrophysiologically in response to visual stimuli. These brain areas can be plastic, but they, to these stimulus, there is an increase in firing, but there is no C-force expression. So the situation again is not is complicated, and I would be very interested in finding this out finally, because this is something which makes all the field contradictory. If you will look at how the engram cells are labeled now by Tonegawa and others, the idea is that you have expression of immediate early genes. On the promoter of immediate early gene, you put something, and this can introduce the channel rhodopsins, optogenetics, drivers, etc., to manipulate memory trace. So this is number one. Number two, you have to bear in mind that C-force is not just a marker of activity. It is a transcription factor which works in the nucleus to change the programs of expression of other genes. It is for something. It is not to mark the cell. We still don't understand it quite well.
So I have rather a superficial question based on what I read when I was a teenager. So I read one of Oliver Sacks' books, and there was a case when patient have some brain injury and he suddenly gained access to all his childhood memories. Right now I have a question. So in case of memory loss, to what extent it is a question of losing the engrams or losing the ways to extract them? So we just can't access them, or is there nothing to access?
The short answer, we don't know. Again, traditional view that the lost memories are lost. But this is some of our mappings of gene expression during single episode of learning in contextual fear conditioning. And each green dot is one cell. And here we can switch this also in hippocampus. Huge amount of cells, about 500,000 of neurons during the single episode of learning in the brain where there are 70 million of neurons. And this is the engram. Now think what it takes to destroy this engram, to find each of these 500,000 cells and harm it. There is no chance that you can do it. You will need to destroy all the brain. That means that memories are lost by expression when we are not able to retrieve. Most probably when just a small portion of the whole circuitry is damaged and it cannot be reintegrated in a proper way. And many people in psychology were thinking that probably the amnesia is not a result of the loss of memory, but inability to access it. which means that experimentally, if a significant portion of neurons of the engram are intact and only disintegrated, it is possible somehow to palpate them, to stimulate them, to reintegrate them back. And we started these experiments and showed that, yes, we can do this, and the memory which has been lost can be returned back. In many different conditions, it comes back. It can be done in mice, it can be done in chicks with the same result. Again, about humans. There is a strange phenomenon which is gaining much more and more attention. I just returned from the conference in Barcelona in July, where there were many speakers about this. It is about very strange thing that the patients with Alzheimer's disease, which have complete loss of memory, they cannot recognize themselves. They are not able to recognize their relatives, their family, where they are, complete loss. Briefly before they die, few hours before the death or 2 weeks before the death, there are two types of this. They strangely recover all the memory. They recognize again all their family, they recognize their past, they start to speak, they remember their past, they are normal, and then they die. It was called terminal lucidity. It has been known from the ancient Greek times. It has been described by medical doctors at the beginning of the 19th century and then forgotten. But caregivers in the clinics with the patients with Alzheimer's disease observe it regularly. They are just afraid of telling this. And during the last several years, they started to tell this, and medical doctors started to tell this. And many papers start to appear with the statistics of this event. It is very regular. It was renamed paradoxical lucidity to be more politically correct instead of terminal lucidity. And there is a lot of things going on here, including the possibility of thinking of how it is possible then to recover these memories by optogenetic stimulation, etc. Psychedelics. There is a large program of NIH in many clinics in the United States to start by medical doctors to explore this phenomenon. Спасибо.
Thank you much for the talk. So actually, I wanted to ask about those methods. And according to the some molecular data that you have described, which methods of treating those reconsolidations, diseases like neurodegenerative diseases are more effective? I mean, behavioral or pharmacological approaches?
We didn't try behavioral because for these, it requires the chronic models. And this has to be done in collaboration with the medical doctors, with the real patients. And it is a longitudinal study. It is a long-term study because you have to accumulate comparison of the patients who are kept in the homeostatic environment, stable and without novelty comparable to others, and to see the dynamics of the clinical picture. We did it pharmacologically, showed some data. We have more extensive. We started to look if our hypothesis is true and we can prevent, make protectors of active memory by preventing its labilization through influence on the NMDA receptors, then not only MK21 and memantine, but other NMDA antagonists, some of which of them are used in clinical trials or in clinics, but were never tested in these neurological conditions, should have the same effect. And in experiments in chicks, we showed that several of them are able also to protect the memory impairment during reconsolidation.
Thank you for your wonderful speech and nice presentation. I have a question I want to ask you that. Firstly, you talked about molecular biology. You mean, I mean that with the help of the bioinformatics, you can to read the gene expression. So in your work that in last year or in this year you published, there is one publication of yours, your student named Akaterina or some people in MSU that you put the mice in a fear box and use electric current to stimulate the mice, and you prove that one special protein to take part in the process of the memories. And in China, my college, they did the same experiment. They recorded the neural activities in the hippocampal area, but we not observe something special. Like the firing rate, there is no something special. Maybe it's just because our data quality is not really good. Then my question is, as your best on your experience, how are you thinking about the neuron to help people to do the memorize, use the energy theory, or to help with the help of the electrical way to transfer the information?
I'm not sure that I got your question, and I don't want to take the time from the audience. What if we will talk later on, and I will try to answer it?
Okay, thank you.
Thank you again for your presentation. I'm not sure if we have much time left for the... Okay, then I will try to be concise. I have actually two questions. One question is, I'm not sure that I got it right, but do the mechanisms that you mentioned within the presentation, do they refer only to the episodic memories, or are the same mechanisms in semantic work for semantic memories, procedural memories, and others? And another question, or maybe a request that I have, if you could give some snippets of how to restore disrupted memories, that would be also great, because it sounds interesting, and I would like to know if it is possible to make it.
All right. I will start from the second. It is easy. We use the reminder procedures, behavioral reminder. There are two ways to do this. You can do it pharmacologically, and it has been shown not by us, but several drugs which increase the arousal can lead to recovery of impaired or lost memories. Probably this is something like what happens during terminal lucidity, when the organism is dying and is desperately striking to survive and release a lot of hormones and catecholamines acting on the nervous system. It, as I told, reintegrates the bits of the old memory so that they become again reintegrated and accessible. So one is pharmacological. The second, which we used, which is more specific, is to present the amnestic agent, animal in our case, pieces of the situation which were in the situation of memory acquisition. It is not new learning, because there is no complete learning episode, but some elements, order, some parts of the environment, some taste, etc., which were used there. without any other associative learning can lead to recovery of memories which before that were looking to be lost.
Thank you.
Yes.
Thank you.
The first question, remind me please, is about what?
Yes,
Different types of memory and how they are consolidated. No, they are not consolidated in the same manner. Most of the data which we have on the dynamics of memory consolidation molecular mechanisms are about contextual or episodic like memories or associative memories, which are formed during a particular episode of associating one event with another or giving a complex stimuli like a context. It is easy to start from this to measure what is going on in the brain, hippocampus, in cerebral cortex, in amygdala, etc. It's more difficult for semantic memories. Semantic memories, first, they are not acquired during one episode, they're cumulative. In order to get a new concept of something for the animal or for the human, you need many events on the, and you don't know at what time to interfere with this. So potentially they are going through the same mechanism, which is repeated many times, and for us it is difficult to target it. With motor memories, yeah, more or less similar to contextual memories, protein synthesis dependent, gene expression dependent.
Thank you.